so I'll introduce myself. So as I mentioned at the beginning of the webinar, I'm the Chief Scientific Officer at biocrates, and many of the people in the audience, I'm sure, already know me from numerous activities where I share information on metabolomics, but also multiomics through our webinars, on LinkedIn and at conferences. And so today I'm going to continue something I started earlier this year. I'm going to continue talking about 5P medicine, and I'll explain in a second what that is. But given the topic of this webinar today is going to be multiomics for 5P medicine. So, sorry, I have to click in the right screen. So yeah, so we'll discuss the multiomics for 5P medicine and continue to the next slide if my computer wants. Yes, now it's working, sorry. So as I mentioned, I will introduce the concept of 5P medicine, and then we'll look specifically at what metabolomics brings to multiomics studies. We'll also take a look at how to do multiomics data analysis, and then I'll finish with a few case studies, examples of how omics have been brought together in multiomics analyses. We've had a beautiful example of that in the previous talk, but I'll touch on different studies where different outcomes have been achieved because the scientists have combined omics together. And then we'll see how we have the future of health at our fingertips, really thanks to the added value of those new molecular tools. I mean, they're not exactly new anymore, but compared to the way we've done research in medicine for a long time before that, they're relatively new in the last couple of decades. But so let's dive into 5P medicine, which is a concept that I've started to introduce earlier this year in our microbiome webinar, and I want to spread the word about this because I think it's a really nice concept to group together a lot of the applications that can be made with omics in general and with metabolomics, and that can really help us to get a better medicine in the future. So if we think of our healthcare systems, and especially in the Western world, these systems have focused on fixing what's broken. And this is a concept that is discussed. So there are a lot of people already see just in our own experience of these healthcare systems and of medicine. We're really good at fixing broken bones. We're really good at treating infection for the most part. We understand how these mechanisms work, and we've developed good strategies to fight them. But on the other hand of that, we have a lot of chronic disease that are rising worldwide. And this is not even just in Western countries anymore. This is worldwide in most countries. You see diseases like type 2 diabetes and obesity, like dementia, and other types of neurological diseases, mental health disorders, and several types of cancers that are not only on the rise, but we don't seem to be able to slow down this rise or to cure any of these diseases, which is why we consider them chronic. And so if you're interested in a system, like in analyzing this system and seeing how molecular tools and modern medicine can bring us from a sick care to a really healthcare system, I encourage you to read a book by Leroy Hood and Nathan Price called The Age of Scientific Wellness. This is where I first heard about 4P medicine. So I will explain what 5P medicine is in the next slide, but the idea is really to gather these concepts that harness the most advanced parts of technology and research and infuse that into medicine so that we have better tools and we go not just to fix things that are already broken, but for example, prevent disease and also find ways to cure diseases that are not just fate, and once you have it, you just have to wait for it to progress more or less slowly, but really find better ways to target these chronic diseases. And so this is this sort of new concept of healthcare where molecular measures of health, so in this case omics, often coupled to high resolution measures of lifestyle, and more this is happening. So this can be through wearables or any type of monitoring that we can have. We talked earlier about stress, for example, but other variables that are going to influence our health, not just over a couple of days, but throughout our lives. So we know that our diet, for example, influences our health, and we see this progressively influencing our metabolome as well. And you see that with lots of different factors that are known to be very important risk factors for these chronic diseases I discussed before. So combining molecular measures with omics and measures of lifestyle together is going to completely change the way that we do medicine in the future. And it's going to enable a new vision of healthcare where we can ensure that people stay healthy rather than waiting for them to fall sick. And then, prevent disease from happening, or for people who are already ill to find new strategies to treat and cure them, and also monitor patient response. So those 5 Ps that make the context of that 5P concept are preventive, predictive, precision medicine, participatory medicine, and population-based medicine. And I will come back to that in the next slide in more detail. But you see here the 5 Ps in one place. And when you use one or more of these approaches, then you start to really have a space where you can harness these molecular tools, these omic tools, and also start to build new approaches to medicine that are really going to have an impact in the future. So how do we support the 5 Ps in research today? And what is the place of metabolomics in 5P medicine? These are the 2 things that I'm going to dig deeper into in my talk. So this is, at the moment, my picture of the 5P medicine, especially from the point of view of metabolomics. So you can think of different tools, and you can think of ways that multiple omics can contribute to each of the tools that are presented here. But metabolomics is especially well suited for all of the things that you see written here in ochre golden letters. For preventive medicine, for example, metabolomics is a wonderful tool to calculate risk scores, or to identify proper, like the best lifestyle intervention or dietary intervention, and then also monitor the effect of those interventions. Metabolomics is a beautifully sensitive, responsive tool to build this kind of tools. In terms of predictive medicine, the typical thing that people build here are biomarkers, and metabolomics is really well known for being a great omic to use for biomarker discovery. But we can also build methods using metabolomics to do better development of drugs, and also, as we've seen in the previous talk, also to have better prognosis and better suited therapies for groups of patients, but also for subgroups of patients. And this is where it goes more towards the precision medicine aspect, with better stratification tools, especially with metabotyping. Metabotypes are very similar to stratification, but it's when you use metabolomics to get a broad screen of your patient's biology, and then you use that to cluster groups of patients or individuals, and those clusters are going to be your metabotypes, and they're going to have very different metabolic profiles, so they can be driven by the same metabolites but in different directions, for example. This is going to give you a very refined picture of the disease that you're looking at. And we know that a lot of the chronic diseases that we have problems with at the moment have a very heterogeneous presentation. But the clinical endpoints that we have today are not always refined enough, or don't have a high enough resolution to really separate those subtypes. And metabolomics is really a tool that's great to do just that. Participatory medicine is probably the one that people know the least at this stage. I hope that by the time I'm done talking about 5P medicine, it will be a more broadly accepted notion. But I think we all know about aspects of participatory medicine when we think of how to make medicine accessible to more people. There are many populations that don't have access to a hospital easily, or that don't have the possibility to go to the doctor very often. And there are strategies that exist, for example, at home sampling, where you can collect your own blood at home, often in a dried form. You can collect other types of samples, and they can be they're stable enough that you can still measure metabolomics, or at least parts of metabolomics in them, and they can be shipped in the mail, and they enable more people to have access to medicine, and also they enable to broaden a bit the spectrum of where we apply our research, because it's really interesting to look into those sampling devices. But of course, when you receive those samples, or also when you measure metabolite levels in an individual, you need to have a reference range to know if this is a normal concentration or not. And so for that, reference range is based on quantitative, quantification of metabolites, for example, in the blood or in other matrices, are necessary in part to enable this participatory medicine aspect. And the last P in the 5 Ps here is population-based medicine. And here, this is where I will focus today, because this is where I see the most obvious application of multiomics, although, of course, we've seen a beautiful example in the previous talk. Multiomics is a great way to work up towards predictive medicine. It's extremely powerful for preventive medicine and completely applicable to all of the 5 Ps. But I only have a few minutes, so I will focus on the use of multiomics here in the context of population health for most examples. And as we kind of touched in the discussion earlier, metabolomics especially is a very sensitive measure of everything that's not genetic, and I will discuss that in more detail in the next slide. But environmental variance or variance from environmental sources is something that you don't really have access to with genetics. Other omics can be more sensitive to it, but really metabolomics is especially sensitive to it and can really bring a lot, especially in population-based medicine. So let's dig deeper into what metabolomics brings to multiomics studies. First, a bit of background. So I don't know if this is obvious to everyone, but I think it's worth mentioning that omics layers were artificially separated by us scientists. So if you look in the cell or even in the blood, metabolites work together with gene products. They're constantly in contact with proteins, with RNAs, with the DNA, and our cells and all the structures of our body are made from this mix of molecules that have very different properties, and this contributes to signaling, this contributes to all sorts of mechanisms. But as scientists, when we wanted to measure them, we decided to separate them based on their structures, because it made it easier to kind of purify them and then go look deeper into the genome of someone or go look into their transcriptome. But really what we've done is we've completely destroyed the system. And when we do multiomics, we're now building it back together. And so this is quite a challenge, but it's also really fascinating. And personally, I started my scientific career as a scientist, as a researcher, bringing these layers together. And so I know that is both extremely fun and fascinating and can be a little bit frustrating sometimes, and definitely a lot of work. And so I'm really excited to discuss this topic today, because it really brings me back to some of the things I really love about science. But I also want to point out that in a multiomics approach to health and disease, metabolomics is really a unique readout. And we've heard this expression. I think it's a really beautiful way that Rosario Spencer put it in her previous talk, that it's a phenotypic endpoint readout. So a lot of people I talk to know about the genome very well. They're very used to genetics, and now they dabble in epigenetics, transcriptomics, even proteomics, and they find easy ways to connect those things together. But a lot of people come from, the Human Genome Project and like understanding from our genome who is going to get sick, what are going to be the best treatments for those people. And we know that from that, we only get about 10 to maximum 20% of the variants that exist in the population for a given disease. So that means the rest of that comes from not genetics. So that comes from the environment, the way we live our lives, the things we're exposed to. And the beautiful sensor of that is the metabolome. So combining genomics and metabolomics for me is a beautiful starting point when you want to have a global picture of what is driving the rise of a disease in the population. From the genome, you get the genetic variants. From the metabolome, you measure the variance that comes from the environment. And both of those are very well compatible with all the other omics as well. And what you can do in the end is you combine genetics, possibly with intermediary effectors of the genome of an individual and the measure of the phenotype. And this is your multiomics analysis. So let's look at a couple of, before we go to the examples, another interesting addition brought by metabolomics is that metabolites are really quite active molecules. And they really deeply influence the other omics. Genetics, maybe not as much, unless you have a mutation, which can come from, for example, an inflammatory environment that can be heavily driven by the metabolome, but that's a bit too detailed. But if you look in the first yellow box here, metabolites regulate the transcription to RNAs and the translation to proteins. One of the main drivers for that is that many metabolite classes are ligands for transcription factors. So if you take bile acids, for example, it's been known now for almost more than 30 years, that bile acids are not just those molecules that help us to absorb fat, but they're actually ligands to several transcription factors and receptors, like the FXR receptor. And we know how transcription factors work. They're going to bind their ligand. The ligand can give different types of messaging. It can say switch on the gene or switch off the gene, and it's going to have that effect on hundreds of genes in the genome. So if I'm a metabolite and I'm able to bind actually several types of transcription factors, I actually can have a really quite deep effect on the transcriptome, which I'm going to see in transcriptomics and I'm going to see in proteomics, in addition to the genome of the person, which is, of course, independent of these effects. Also, indole derivatives, for example, are very well-known activators of the AHR receptor, for example. In addition to that, when we look at modifications of the DNA, for example, with epigenetics, or when we look at post-transcriptional or post-translational modifications on RNAs and proteins, these take metabolites as the matter that is being added onto the other molecule. And certain mechanisms can also be regulated by metabolites, for example, through the action of the transcription factors. So you see that in addition to bringing this measure of the environment, you're also actually measuring molecules that have a deep impact on the other omics. So you can really, in terms of interpretation, you have many angles where metabolomics can help you. And so one of the examples I'm going to detail in this presentation is how combining metabolomics with GWAS, so with genome-wide association studies, brings us enormous added value. So GWAS is basically the exercise of taking the genome of a large number of people, usually in a relatively large population. We started with a few dozens, a few hundred people, and now we're at GWASs that have like millions of people almost. And you combine that with a measure of the phenotype, so usually with the clinical markers of the disease. And the associations are relatively weak, and this is where you get this about 10% variance that is explained by the genome. And when you add the metabolome to this, you can build strong associations in both of the other directions. So the metabolome is going to be strongly associated to the genome because, of course, if you follow the typical cascade, the genome is going to influence which RNAs we have, which proteins we have, and how we handle metabolites. So there's going to be an association there. But it's also going to have associations to the phenotype because of the other side. So because the phenotype is influenced by lifestyle and environment, and so is the metabolome. So when we combine GWAS with metabolomics in what is called mGWAS, then we can establish functional relationships very easily. We can find phenotype converting variations, and we can qualify associations that would have been too weak with the disease using genome alone, but using the metabolome, we can kind of reveal associations that exist there. And this is so, especially for people who have already GWAS data and are maybe thinking of doing metabolomics, this can be quite an easy way to add on to an existing cohort or to an existing study by running the metabolome and then doing an mGWAS study to find new associations. So this is what was done by the group of Carsten Suhre and multiple scientists now in the world. But I mentioned Carsten Suhre here because he was one of the leading scientists in the first team that made the first GWAS, which was already in 2008. So this has been going on for a while. And it's a really beautiful example and a really clean example of how this works. So they had a cohort that was not an enormous one. It was still 2008, so we were not at the stage now where we have millions of people in a single study. But already with a few hundred people, of course, using metabolomics, they had some significance or associations with lipids that were associating with SNPs for a gene that is related to lipid metabolism. But what was really interesting here is that they made the ratio of the substrate and the product of the enzyme that was encoded by this gene, and then they basically exploded the p-value of the association. I'm sorry. Oops, I have to go back. Of the association here. So this is what you see in the little table at the bottom. And they could explain a lot more of the variance, because even though they combined two metabolites that were not so strongly significant, especially the second one, bringing them together really kind of revealed the relevance of that gene in the whole study. So I'm not the type of person who does this kind of analysis, so I'm explaining this really poorly. So if you want to learn more about this, please check out the Metabolomics episode. So the Metabolomics is my podcast. And so one of the first episodes that we published was with Carsten Zure, who is this researcher I was talking about, where he explains beautifully this study, and he explains beautifully also the added value of combining genomics with metabolomics in this type of study. So please either scan the QR code here or go to themetabolomics.com, and there you will find this episode on the page. So for people who are interested in GWAS, what we show in this slide is really that you have, this is already a couple of years old, but you see like there are a few thousand studies doing GWAS. Of course, it's a very popular approach, and it's very effective to look at the effect of the genome or the part of the genome in a certain population or disease. But when you add the metabolomics to it, so when we go to the mGWAS level, you can really increase or like have a p-value gain by adding the metabolome to your data set. And when you add to that Mendelian randomization, which is something that is more and more added to GWAS studies to understand not just associations, but also causality. Are the metabolites driving the changes, or are the genes driving the metabolite changes? So yeah, it's really a very powerful tool to use in combination with GWAS with genomics. And how do people do this multiomics analysis? So I'm gonna do a very like superficial view of this, because we'll get a lot more insights in the next talk. But one thing that's really important for me is this kind of dichotomy between an automated versus human-made analysis of the data. And I'm someone who did a lot of things by hand, and as a human being, I love doing this. And I think a lot of bioinformaticians also enjoy like having their hands in the data and really playing with it. But as we heard from the previous talk, and as we will definitely hear in the next talk, you need to build pipelines in order to have a structured approach to things. And the completely opposite aspect of that is using software. Like there are a few pieces of software, especially online, that you can use where you throw your data in it, and then you just it just runs the numbers a bit, but you don't have so much control over it. And for me, the sweet spot is somewhere in between, where you have a pipeline that has been curated by a bioinformatician that kind of fits or perfectly fits actually your data sets. And then you can also get the curation from the bioinformatician. So by building those pipelines, you can considerably reduce the time needed for the humans to work on it, and then you can spend the time curating the results and making sure that everything is in line. So this is the type of multiomic analysis that we offer at Biocrates with the support of Viewgene. And so Viewgene is a company that we will hear more about in the next talk. And Viewgene has built beautiful pipelines for multiple types of omics, including, of course, metabolomics. And they combine these different pipelines based on the needs of the customer to make a tailored multiomics approach to each data set and provide the results. So if you're interested in this approach and learning more about this, you will hear more about it in the next talk, but you can also go to the Biocrates webpage to learn more about it. So I'm a little bit short in time, I realize, so I'm gonna go relatively quickly through the case studies, and please don't feel like you have to read all the text. But each case study brings something specific. The basic level is you combine omics, you want, especially like with genomics or with epigenomics, you want to find new associations. That's really one of the beautiful ways to use metabolomics for this. So here we have an example of a cohort with, I can't remember, yeah, a bit over 3,000 individuals. Exome-wide sequencing was performed, and metabolomics was measured with the absolute IDQ P180 kit. And this is just a typical example of finding associations you just wouldn't find by using the genomics alone or the epigenomics alone. There are other cases where you can enhance pathway discovery. So in this case, you can conduct the epigenetics, you can conduct the metabolomics, and you may have for example, bile acids that are part of your metabolomic panel, and you will have a lot of bile acid related genes that will be covered by the DNA methylation array, of course, but it's only when you combine the two that this pathway comes to the surface, and that you see that you actually have loci that are related to bile acid metabolism that associate really well with taurine, which is a metabolite that's related to the processing of bile acids by the host, by humans, and also by the microbiome. And the last case study is one that I like a lot, because this is a case where the authors started with stratifying a cohort of Alzheimer's patients based on their metabotypes. So they did that thing I was talking about earlier, where you take the metabolome and then you screen, you kind of cluster the individuals based on their metabolome, and those subtypes were then processed to look at the other omics. And this really revealed interesting differences, for example, in acylcarnitine species that were different in the different subgroups. So this is really, for me, a nice demonstration of the power of starting with the metabotype and then digging deeper into the complexity of a disease. So with all of this, I know it's a lot of information, but with all of this, I think we really have the future of health at our fingertips, and omics, and especially multiomics, are a big part of that. So I hope that you learned today about the 5P medicine, that I could convince you that this is an interesting concept that really packs a lot of what's really exciting and promising about omics and about metabolomics for the future of medicine. So take this with you, talk about it with your colleagues, and let's translate omics to make it become the future of medicine together. So thank you for your attention.